What if I told you that a dark red, heavily staining tree sap-like, a substance ancient civilizations actually used for centuries to dye artwork and treat really severe battlefield wounds, is now being tested in zero-gravity space
That
simulations?
sounds totally made up.
It really does. But it's also being used in, high-end clinical skincare trials today. So welcome to the deep dive. Today we are looking into the botanical mystery that is dragon's blood.
Yeah, and it's an incredibly evocative name.
Yeah.
It almost sounds like something pulled straight out of a fantasy novel.
Oh, absolutely.
And because of that mythic reputation, I think it's really easy to just dismiss it as ancient folklore or, snake oil. But the moment you take this red resin out of the history books and actually put it under the scrutiny of modern analytical chemistry, the biological mechanics are just staggering.
And that is exactly our mission for this deep dive. We're taking a massive stack of diverse sources to figure out if this legendary cure-all actually holds up to modern science. For you listening, we are pulling from botanical analyses out of the Indonesian jungles. We've got antimicrobial lab studies from Yemen, a brand new 2025 clinical trial on cosmetics, and an absolutely wild pharmacological study that simulates outer space.
Yeah, that space study is fascinating.
It really is. So whether you are obsessed with cutting-edge skincare, or you're fascinated by how our bodies actually absorb medicine, or you just love a really good botanical mystery, this deep dive is going to fundamentally change how you view natural remedies.
It really bridges the gap between the ancient apothecary and the modern lab. I mean, in a way, very few natural substances do.
Right.
But to truly appreciate how this red resin interacts with human biology, we first need to, strip away the mythology and look at the botany.
Right, because what exactly is it?
Well, one of the biggest misconceptions out there is that dragon's blood comes from one single type of magical tree. It doesn't.
Yeah, it's actually a historical catch-all term, right? It refers to red resins harvested from several totally distinct plant families spread all across the globe.
Exactly. Our sources highlight two radically different environments. First, there's Yemen, specifically the remote monsoon-swept Socotra Island.
Okay.
This is the home of Resina cinnamomum. And if you were walking through this harsh, arid landscape, you'd see this bizarre tree shaped almost exactly like a giant inside-out umbrella.
Or like a massive stalk of broccoli.
Yes, a huge broccoli stalk. And the shape is actually a direct evolutionary adaptation.
Yeah.
Socotra Island gets intense summer monsoons, fierce winds, blistering heat.
So the shape protects it.
Right, the dense, umbrella-like canopy provides shade. It helps the tree capture moisture from the fog, which then drips down the trunk to the roots.
Oh, wow.
And when the bark of this tree is sliced, it secretes a thick, dark red liquid that instantly oxidizes. I mean, it literally looks like the tree is bleeding.
Hence the name. But if we travel thousands of miles east, from the arid deserts of Yemen into the dense, humid rainforests of Indonesia, the botanical source of dragon's blood completely changes.
Completely.
In Indonesia, it doesn't come from a bleeding tree trunk at all. It comes from plants in the Demonarops genus.
Which is an entirely different family of plants. These are actually rattan vines.
Wait, rattan. Like the exact same tough, woody climbing palms that people use to weave wicker chairs?
The very same.
That is so weird.
It is. It's fascinating to think that the mundane material making up a piece of patio furniture secretes this highly prized, biologically active resin. And because the plant is a creeping vine, the extraction method is totally different from the Yemen trees.
Right, it doesn't bleed from the stem.
No, it naturally forms as a brittle, red crust on the outside of the scales of the rattan fruits. Local communities have actually spent generations developing specific techniques to harvest this resin without destroying the plant itself.
Yeah, the sources detail these traditional extraction methods, and they are surprisingly sophisticated. There's a dry method where they simply take the dried rattan fruits, place them in baskets, and physically pound them.
Just bash them until the brittle resin shatters and shakes loose from the scales.
Right. But the wet extraction method, which is used heavily in places like Hoksimawa, is where it gets really interesting. They take the pounded fruit skins and put them into a container filled with water. Then they vigorously spin, rub, and squeeze the plant matter.
So they're essentially using mechanical force to wash the resin off the scales.
And the water turns this deep, cloudy crimson.
Yeah.
Once they filter out the leftover plant fibers and debris, they leave that crimson water out in the intense tropical sun. The water evaporates, and the pure, solid resin precipitates out and settles at the bottom of the container as this highly concentrated paste.
Which is a brilliant low-tech refining process.
It is, but reading through that raised a massive red flag for me. If the sources of dragon's blood are so wildly different, I mean, a desert tree in Yemen versus a woven furniture vine in Indonesia, not to mention other species in China, how do scientists even know what they're looking at in a lab?
It's a challenge.
Like, if someone hands you a vial of red powder labeled dragon's blood, how do you authenticate it?
Well, you have to look for a chemical fingerprint. Our Indonesian sources used a highly sophisticated analytical technique called gas chromatography mass spectrometry, or GCMS.
Okay, GCMS.
In simple terms, this machine vaporizes a tiny sample of the resin, pushes the gas through a long tube, and separates the individual molecules based on their weight and size.
Got it.
So they took 16 different samples of dragon's blood from various towns across Indonesia, representing different rattan species, and ran them through the GCMS. The machine identified 42 distinct chemical compounds within the resin.
Wait, 42 different compounds sounds like an absolute chemical mess.
Yeah.
If you're trying to find a reliable fingerprint for Indonesian resin, how do you isolate one miracle marker out of all that biological noise?
You search for the dominant constant. When the researchers analyzed the data across all 16 geographic samples, they found that out of those 22 compounds, one specific, highly complex molecule was universally present in massive quantities.
And what was that?
It's a compound called Draconis. That molecule is the unique major marker for Indonesian Demonerops resin. If a lab tests a red sample and it's rich in Draconis, it is unequivocally Indonesian. By contrast, Chinese dragon's blood relies on a completely different active chemical marker called lurarin.
So Draconis is essentially the biological ID badge for the Indonesian vine.
Exactly.
And the GCMS testing revealed something else pretty incredible, right? These traditional extraction methods like the pounding and the water spinning yield an incredibly pure product.
Oh, absolutely. The tests consistently showed actual resin contents above 80%, which officially classifies it as super quality.
Wow.
That high concentration is exactly why pharmacologists are paying so much attention right now. We know what it is, we know how to harvest it, and we know how to chemically verify it.
So we have this miracle resin that slaughters bacteria on contact in a lab. You'd think the logical next step is just to crush it into a pill.
You would think so.
But when researchers took this highly pure resin from the forest and put it into a Petri dish, the biological activity they witnessed was startling. Let's talk about the healing power.
Let's do it. So the Yemeni Resina resin was tested against various pathogens, and the results were ruthless.
Ruthless is a good word for it.
When they used a chloroform extract of the resin, it showed massive antibacterial success. We're talking about destroying serious disease-causing bacteria.
Like Staphylococcus aureus, right? Which causes terrible skin and respiratory infections.
Yes, and Micrococcus luteus too.
Yeah.
The mechanism behind this destruction is fascinating. The phytochemical screening reveals that these resins are absolutely packed with flavonoids.
Okay.
Specifically, they're rich in a subclass of flavonoids called anthocyanin derivatives. Plants naturally produce anthocyanins as a defense mechanism. It's basically a form of chemical warfare against invading pests, fungal infections, and extreme environmental stress.
And Draconis, that chemical ID badge we just identified for the Indonesian resin, is a potent anthocyanin derivative.
Exactly.
And that specific compound does heavy lifting in two ways. First, anthocyanins are pigments. So Draconis is what gives the resin that striking, deep, blood red color. But second, it provides massive antioxidant properties. And I really want to get into the how of that. We hear the word antioxidant thrown around in health food commercials all the time, but the underlying chemistry is wild.
It really is.
Inside the human body, you have these highly reactive, unstable molecules called free radicals.
They're unstable because they are missing an electron. So they roam violently through your body, basically desperate to find an electron to steal.
They'll just rip electrons off your cell walls, your proteins.
Even your DNA.
Exactly. And that cellular damage accelerates aging, causes systemic inflammation, and is like a major trigger for cancer.
But Draconis has a very unique chemical structure. It features these complex conjugated double and triple bonds. Basically, it has a surplus of easily accessible electrons.
Okay, let's unpack this. So instead of like a catcher's mitt catching the bad guys, you can think of Draconis as a molecular pacifier.
I like that.
When it encounters a violent, unstable, free radical tearing through your cells, it simply hands over one of its extra electrons. It pacifies the radical, instantly neutralizing the threat before it can cause cellular damage.
That is an excellent analogy. The double and triple bonds allow the Draconis molecule to safely donate an electron without becoming unstable itself. It's just an incredibly elegant, highly efficient chemical trap.
It really is.
And lab tests have repeatedly proven its efficacy as a stabilizing antioxidant and an antimicrobial agent.
Okay, so we have this dark red resin that acts as a molecular pacifier against free radicals and destroys staph infections on contact. The natural question is, why aren't doctors prescribing this as a daily pill?
Right. If traditional healers have been swallowing it for centuries to cure internal illnesses, why isn't it on pharmacy shelves right now?
Yeah. This brings us to a massive, frustrating wall in modern medicine known as pharmacokinetics.
Yes. Pharmacokinetics is the study of a drug's life cycle inside your body. It maps how a substance is absorbed through the gut, distributed through the blood, metabolized by the organs, and eventually excreted.
Basically, the journey of the drug.
Exactly. Because getting a compound to neutralize a threat in a controlled, watery petri dish is relatively easy. Getting it to survive the acidic gauntlet of the human stomach, cross the complex intestinal walls, and enter your bloodstream in high enough concentrations to actually heal you, that is an entirely different game.
And this is where Dragon's blood completely stalls out.
Completely.
The sources detail a pharmacokinetic study on rats, tracking one of the active compounds, Loran B. When the researchers gave the rats a pure, isolated liquid dose of just the Loran compound, it worked flawlessly.
The body absorbed it rapidly.
Right. It peaked in the bloodstream in under an hour, and it distributed heavily to organs with high blood flow, like the liver and the kidneys, which is right where you'd want an antioxidant to go.
But there is a massive catch. Traditional medicine practitioners and, people buying supplements today, they don't consume pure isolated Loran in a synthetic liquid.
No, they consume the raw, natural resin extract.
Exactly. And when researchers gave the rats the raw sap making, absolutely sure the exact same milligram dose of the active Loran compound was present, the absorption just crashed.
Systemic absorption dropped by a staggering 17 times.
Yeah.
Wait, so traditional medicine practitioners who ingested the raw sap for centuries might not have been absorbing much of the active compound at all.
Precisely. The vast majority of it was doing absolutely nothing.
That is wild.
And the mechanism behind this failure is something pharmacologists call the resin trap. You have to remember what this sap evolved to do. It evolved to form a hard, protective crust on the outside of a plant.
Okay.
The raw sap isn't just made of the active medicine. The medicine is suspended inside an incredibly complex matrix of sticky polymers.
Mm-hmm.
And these polymers are heavily lipophilic.
Meaning they're fat-loving and water-hating.
Exactly. The human digestive tract is essentially a watery slip and slide. When you swallow the raw resin, these lipophilic polymers refuse to dissolve in the water-based environment of the stomach and intestines.
So the resin just acts like a stubborn ball of wax.
Yes. And the active healing molecules are literally trapped inside this waxy, sticky polymer matrix. They cannot escape to make contact with the intestinal walls.
Wow.
So instead of being absorbed into your bloodstream, the complex resin, along with all the medicine trapped inside it, is simply pushed through the gut and flushed out of the body in feces.
The irony there is so frustrating. I mean, the exact same sticky, waterproof polymer that protects the active compound out in the harsh jungle, defending the plant from heavy rains and pests, is the very thing that prevents it from working inside the watery environment of the human body.
It is the ultimate proof that natural does not automatically mean bioavailable.
Totally. And if the resin trap wasn't a strange enough hurdle, our sources detail a truly bizarre study that took this pharmacokinetic problem and tested it in the most extreme environment possible.
Outer space.
Outer space. This was the source that completely derailed me. Scientists actually ran a simulation to see how a mammalian body processes botanical medicines in microgravity.
Yeah.
But I have to ask, why even bother testing a terrestrial plant resin in space?
Because space agencies are actively trying to map out the pharmacy of the future. When astronauts are on long-duration missions on the International Space Station or eventually on a trip to Mars, they will inevitably get sick or injured.
Right.
We need to know if the medications we manufacture on Earth will still work when swallowed in an environment without gravity. Digestion isn't just a matter of your stomach muscles squeezing food down the pipe, peristalsis.
Right, the squeezing motion.
Yeah, but gravity plays a massive, invisible role in how fluids mix and settle in your gut.
Because on Earth, gravity pulls the liquid contents of your stomach and intestines down, making sure that the digested material constantly presses against the absorptive walls of your intestines.
Exactly. But in microgravity, fluids behave completely differently. They tend to form free-floating spheres. So the researchers wanted to know what happens to the already stubborn resin trap when you remove gravity from the equation. They simulated microgravity conditions for the test subjects and tracked the absorption of the active compound again.
And the results were drastically worse. Under microgravity, the absorption of the active compound plummeted by another 34.7% compared to the already terrible absorption under normal Earth gravity.
Furthermore, the timeline of the absorption completely fractured. On Earth, the drug hit the bloodstream in one concentrated peak.
Like you'd expect.
Right. But in microgravity, the way the drug moved through that digestive tract was so disrupted that it created a strange double peak in blood concentration.
A double peak?
Yeah. It suggests that without gravity pulling the resin against the intestinal wall, the medicine sort of just floats in the center of the intestine, which severely delays and cripples the absorption process.
That is deeply unsettling when you think about the implications for space travel. But bringing it back down to Earth, if swallowing this resin is basically throwing it away because of the biological trap, how do we actually utilize its massive antioxidant and antibacterial power?
Because we know it works in a petri dish.
Exactly. Modern science has essentially engineered two pathways to bypass the gut. We either use cutting-edge nanotechnology to smuggle the medicine into the blood, or we skip the internal route entirely and apply it directly to our skin.
The smuggling route is still in development, but it is highly promising. Pharmacologists are actively working on using nanotechnology to beat the resin trap.
How does that work?
They isolate the pure active compounds, entirely stripping away those sticky plant polymers we talked about. Then they wrap those pure molecules in microscopic lipid nanoparticles.
So they're giving the medicine a fat-based protective suit, like a VIP biological disguise.
Precisely. Because the active compound is now wrapped in tiny, sophisticated lipid armor that the human body easily recognizes and absorbs, it can bypass the harsh stomach acids and cross the intestinal wall directly into the bloodstream.
Oh, that's smart.
It entirely circumvents the natural trap the plant originally set.
Which is brilliant engineering. But while those nanotech pills are still being refined for the pharmacy, the second solution topical application is already proving to be a massive triumph.
Oh, yeah.
And this brings us to that fascinating 2025 clinical trial on Indonesian dragon's blood.
This trial is the perfect intersection of ancient history and modern clinical validation. We know that thousands of years ago, people rubbed this sap on severe wounds to prevent infection. Well, modern researchers wanted to see if the immense antioxidant properties of the resin could be weaponized into a modern daily sunscreen.
Sunscreen?
Yeah. They formulated a topical cream-dubbed Formula F3 using refined Indonesian dragon's blood and tested it on 30 human volunteers over a strict 30-day period.
And this was a rigorous dermatological test. They use high-tech skin analyzers to measure the physical barrier of the skin and specialized UV cameras to physically visualize the UV protection blocking radiation.
And the results were nothing short of phenomenal. Under the UV camera, the Formula F3 sunscreen maintained 100% UV protection for the entire 30-day testing period.
100%?
Yeah, it completely matched the protective capabilities of commercial sunscreens that rely on heavy metals like zinc oxide or synthetic chemical blockers.
I really want to focus on how a plant resin deflects UV radiation because it ties back to the botany. You mentioned earlier that plants produce these anthocyanins as a chemical defense mechanism.
Right.
Well, out in the harsh sunlight, a plant can't put on a hat or walk into the shade. The anthocyanins in the red resin literally act as a natural biological sunblock for the rattan vine,
absorbing the destructive photons of UV light before they can mutate the plant's DNA.
Exactly. So when we extract that resin and formulate it into a cream, we are essentially borrowing the plant's evolutionary UV shield and painting it onto our own skin.
It's exactly the same mechanism repurposed for human biology. And crucially, it provides this barrier without the risk of synthetic side effects.
That's huge.
The irritation test was a critical component of the 2025 trial. Over 48 hours of constant observation, tracking erythema, which is clinical skin redness, and edema, which is swelling, the dragon's blood sunscreen caused absolutely zero irritation.
No redness, no swelling.
None. It was universally safe for regular daily application.
But it wasn't just acting as a passive shield, was it? The formulation actively improved the overall health of the skin.
Yeah, because the researchers didn't just use the raw resin, they engineered a synergistic blend. They paired the dragon's blood with 4% niacinamide, olive oil, and moringa seed oil.
And the synergy is where the magic happens.
Mm.
The dragon's blood provided the heavy-duty UV blocking and acted as that molecular pacifier, neutralizing the free radicals generated by environmental stress and pollution.
Right. And meanwhile, the accompanying oils and niacinamide locked in the skin's natural moisture and repaired the skin barrier.
The clinical data backs up that synergy beautifully. After the 30-day trial, roughly 2/3 of the users experienced a measurable, significant increase in their skin's brightness.
Wow.
The moisture levels on their skin dramatically improved, while excess, unwanted oil production actually decreased.
That's incredible.
So what does this all mean? Let's step back and look at the sheer irony of this journey. We started this deep dive looking at a dark red, heavily staining tree sap, a substance that ancient civilizations literally used to dye canvases and paint pottery red.
Yeah.
And now, in 2025, rigorous clinical trials are proving that rubbing this deep red dye onto your face is actually the scientific secret to getting clearer, brighter, and perfectly UV-protected skin. It is a wildly counterintuitive paradox.
It is a paradox, but I think it perfectly illustrates the evolution of how we understand natural medicine. We don't have to discard traditional remedies as mere fairy tales.
Right.
The ancient healers were absolutely right that this resin possessed incredible healing properties.
Huh.
But we also can't blindly assume that these plants work perfectly in their raw, natural form when swallowed.
Exactly.
They have used the rigorous tools of modern science, from GCMS chemical fingerprinting to advanced clinical trials, to isolate what actually works, understand the microscopic mechanics of why it works, and engineer intelligent solutions to bypass the biological traps that nature originally set.
From the windswept, umbrella-shaped trees of the Socotra Island desert to the pounded rattan vines of the dense Indonesian jungle, we have traced the chemical ID badge of Draconis, watched molecular pacifiers hand over electrons to neutralize destructive free radicals, and discovered how the stubborn lipophilic resin trap completely outsmarts the watery environment of our digestive system.
That's quite a journey.
It is a massive testament to the sheer, complex chemical brilliance hiding inside the plants we walk past every single day.
It truly is. And, as we conclude, there is a lingering implication from our stack of sources that goes far beyond skincare, ancient history, or even botany.
What's that?
Think back to the microgravity study we unpacked. It's simply removing the invisible physical force of gravity alters the way fluid mixes in the gut, drastically reducing the absorption of an ancient plant compound by nearly 35%?
Yeah.
What does that mean for the entire catalog of modern medicine when humanity eventually pushes further into the solar system? If the lack of gravity inherently breaks human digestion, will the space agencies of the future on Mars or on a deep space station be forced to completely reformulate and re-engineer every single pill, painkiller, and antibiotic we currently take for granted here on Earth?
That is an incredible, unsettling thought to leave on. The very physical environment we live in